Histology applied to tissue engineering is an essential foundation for understanding the structural and functional organization of tissues, as well as the cellular processes that ensure their maintenance and regeneration. The cell, the basic unit of life, differentiates and organizes itself, forming tissues that, in turn, integrate into organs and systems. This process involves regulated mechanisms of differentiation, division, and cell death that ensure tissue equilibrium. In this context, the cell cycle acquires central relevance in tissue regeneration and repair processes. Multicellular organisms originate and are maintained through complex sequences of cell divisions, regulated by specific phases that determine proliferation, differentiation, or quiescence. Likewise, the interaction between cells and the extracellular matrix plays a decisive role in tissue regulation. The matrix, understood as a three-dimensional network of proteins and structural molecules, not only provides mechanical support, but also actively participates in cell signaling processes through a dynamic reciprocity model. This interaction regulates gene expression, differentiation, migration, and cell survival, participating in phenomena such as embryonic development and wound healing. Therefore, tissue engineering requires a deep understanding of histology to reproduce suitable biological microenvironments, avoid adverse immune responses, and promote the functional integration of cultured tissues.